How is it that we're able to send signals from one part of the body to the next? How can our fingertips sense something and send that signal to our brain? How is it that our neurons encode messages to send them throughout our body so that we can act in our environment, respond to our environment, and perceive our environment? Welcome back to Neuroscience, and today we're going to be discussing just those questions. And importantly, we're going to be discussing the action potential, which is the most basic part of every signal sent through our body. To answer all these questions, we need to understand one thing, and that's the action potential. Now, an action potential is the way a signal is sent from the axon of one neuron all the way to the dendrites of the next. Let's just say, for instance, you're chilling, you're happy, and then all of a sudden a fire is ignited underneath your hand. How does the signal get from your hand all the way to your brain so that you can realize, oh my gosh, that's hot, and withdraw your hand? Well, like we said before, again, this signal has to travel from one neuron to the next, moving down the axon until the signal is delivered and triggers another action potential in the postsynaptic neuron. Now, if you want to get really specific, this signal is reflexive. So it's not going to go all the way up to your brain. In fact, it'll just go to your spinal cord, and that can trigger the response. Once things get into our brain, it gets really complicated. We start thinking about how we're going to act. And so for something like a reflex, where you're withdrawing your hand from something hot, you don't want to be thinking about it too much. Either way, action potentials are being sent all along the nerves in your arm, all the way back to your bicep in this case, that's going to contract and pull your arm away from the fire. To understand how these action potentials work, we have to understand that there's different ion concentrations inside and outside of the neuron. So for instance, there are high concentrations of sodium outside the neuron compared to inside the neuron. Chloride is also higher outside, and potassium is higher inside the neuron. Additionally, the proteins our body has inside these neurons are typically negative, and so we have a negatively charged neuron. So this is how we maintain the resting potential. First of all, we have a membrane that's impermeable to these anions, and so we already start with a negative charge inside the cell. Secondly, we have movement of potassium and chloride through leak channels. And so chloride being negative is going to want to move out naturally. Potassium being positive is going to be pulled in. Lastly, we have the also important sodium-potassium pump, which pumps out 3 sodiums for every 2 potassiums that are pumped in, maintaining a gradient. And this gradient keeps the neuron at around negative 70 millivolts. So to start off with resting potential, we are already at a negative state. Now on every axon, there are channels. And we have the sodium channel and the potassium channel, which at rest would both be closed. These channels are voltage gated, so that means that the neuron has to approach a certain voltage before they open, and that is the threshold of around negative 40 millivolts. So to get to this point, there has to be some sort of stimulus, and the stimulus can cause the neuron to become more positive on the inside. Now, if the neuron does not surpass threshold, then it will fall back down to rest. If, however, the stimulus is strong enough to bring the neuron above threshold, an action potential will be triggered. At this point, what we have happening is the opening of our sodium channel. When this happens, sodium from the outside can flood into the cell towards the negative charge. And so the charge becomes more positive. This is called depolarization, and At this point, the cell is in an absolute refractory period where no more signals can be triggered. At the same time that the sodium channels were triggered to open, the potassium channels were also triggered to open. They open slower, however, so this is why we have a curve at the top of the graph, because potassium begins to efflux, causing the neuron to fall back to a negative potential, becoming more negative and repolarizing. So overall, we saw that the neuron depolarized, followed by a repolarization, to the point where it will actually hyperpolarize. After hyperpolarization, the sodium and potassium channels are once again closed. To restore the resting potential, the sodium-potassium pump kicks in and pumps sodium out of the neuron and potassium in to come back down to negative 70 millivolts. Now, the way that this travels down the axon is that when the sodium begins to flood into the axon, the whole inside of the axon is negative, so these positive ions are attracted down the length of the axon. This triggers more action potentials as the other parts of the axon come up to threshold. Of course, when this happens, the sodium channels open further down the axon, and so we have action potentials all along the length of the axon. And that is how the signal goes from the soma down towards the dendrites. Now, you might be wondering how this signal even starts in the first place. The important thing is there's all sorts of receptors gated in different ways that will be triggered due to different stimuli. So how are these signals even starting? Now, it depends on the sensation. For touch, there are different receptors for pressure in our skin, thermoreceptors in our skin as well, which are actually these weird kind of hand-like shaped structures, and so when it gets hot they heat up and expand, and that causes channels to open and kickstarts the depolarization. With our eyes, there's proteins that help the process, including rhodopsin, and this triggers channels to open. At the end of the day, what's important for this talk is that what's happening is we're triggering these cation channels to open, so cations being positive ions, and then these positively charged ions can flood into the cell, causing depolarization, bringing us up to threshold. Once we get to threshold, our sodium channels can open, our potassium channels slowly start to open, but in the meantime we have all this sodium flooding in. Once our potassium channels get open, potassium can leave, causing the curve, and then we fall back down to rest. Alright, that's it for today. I hope you enjoyed our first Neuroscience ABC video. If you have any questions about this video, leave them down below. I'll be sure to get back to you as soon as possible. Any comments, any suggestions for future Neuroscience ABC videos, or any NeuroSciQ videos in general, leave them below. We always look forward to hearing from you. So, you know what to do next. Make sure you like this video, comment, subscribe, turn on notifications if you haven't done so. What are you waiting for? And I hope to see you next week back on this channel. See you then.